activating input. Because of the redundancy in endoderm specifications, removing SKN-1 produced a partial penetrant loss of gut phenotype. Because when SKN-1 is eliminated, all embryos are arrested due to misspecifications of MS. However, fractions of the embryo will still develop gut because of the additional activating input. We want to just understand the genetic regulations of this bistable switch, but because SKN-1 loss of functions is lethal, screening for its enhancer or repressor can be very tricky. We therefore turn to a natural wild isolate. Each of these dots represent a unique haplotype. We did a genetic screen to look for natural variations in the requirement of SKN-1 and early endoderm development. In this experiment, we knocked down SKN-1 in 97 wild isolates and scored for gut differentiation in the absence of SKN-1 and observed a broad variations. In some strains, SKN-1 is absolutely essential for gut development, and in other strains, the gut can largely specify and differentiate even in the absence of SKN-1. And N2, everyone's favorite strain, sits right in the middle of the spectrum. I wanted to add that we have confirmed this results with chromosomal mutations, showing that these variations reflects differences in the GRN input per se and reveal surprising plasticity in the endoderm GRN. Using quantitative genetic methods, we found several genetic regions, or QTL, including 2 in chromosome 2 and chromosome 4, that may explain the SKN-1 variations among the wild isolates. And the allelic effects are shown here. Therefore, we conclude that accumulations of cryptic genetic variants drive the evolutions of the GRN. I'm skipping over a bunch of data, and you will just have to take my words for it, that we found that SKN-1 and Wnt can partially compensate for each other, that in strains with strong SKN-1 requirements are less dependent on Wnt and vice versa. So far, I told you that variations in the developmental input is driven by accumulations of natural genetic variants and wild isolates. But that's only part of the stories, as I will tell you in the remaining time that we found a heritable epigenetic effect that also influenced gut differentiation. To study how SKN-1 phenotype is inherited, we selected 2 strains with very different SKN-1 phenotypes, high strains that produce a lot of embryos with gut, and the low strains that virtually never make gut when SKN-1 is removed, and perform reciprocal crosses with them. First, we observed a very strong maternal effect, such that the phenotype of the F1 followed that of the mother. For the red bar, a low strain, MY16, was used as the mother, and for the blue bar, JU1172, a high strain, was being used as the mother. This is expected. As I mentioned earlier, SKN-1 is a maternal effect. This non-reciprocal effect quickly disappeared in the subsequent generations, as one would predict, as the maternal effect is diluted. And the progeny from both directions showed an intermediate phenotype due to the multigenic nature of the trait, and this experiment was performed with well-fed parents. To a surprise, if the parents were starved as dour before the cross, the non-reciprocal effect persists for at least 10 generations. Crosses with high mom stays high, and crosses with low mother stays low. I wanted to stress that these guys, the progeny, had not experienced starvation, only the parents that started the cross did. That is, metabolic stress in one generation can trigger persistent parent of origin effect, POE, in gut development. We started to explore the possible mechanisms of POE, starting with mitochondrial inheritance. Mitochondria are maternally derived. If starvation triggers changes in the mitochondria, which that get passed on and affects SKN-1 phenotype, It may then explain the POE we observed. To investigate whether maternal cytoplasmic content, including mitochondria, could influence SKN-1 phenotype, we use a genetic trick to create a hybrid with mitochondria from one strain and nucleus from a different strain. In wild type, when two pronuclei meet after fertilizations, we get equal segregations of paternal and maternal genetic material. However, in GPR1 overexpression background, the pronuclei separate before fusion, creating a mosaic animal. And we know that the germline is derived from the posterior P2 cell, so that the next generations will have pure paternal genome, but maternal mitochondria. If starvation causes changes in maternal mitochondria that influence SKN-1 phenotype, we would still expect to see POE in the progeny from this mutant. However, the phenotypes of the progeny that contain paternal nucleus but maternal mitochondria is indistinguishable from that of the father, regardless of the nutritional status, showing that POE is not encoded in the mitochondria. To provide a complementary test, establish the cross like I did before and kept back crossing the progeny to the paternal strain. This way, I keep the mitochondria and exchange out the nucleus bit by bit. If mitochondria do play a role in POE, we should still see POE last despite the back cross. However, despite the strong POE we saw initially, this effect quickly disappeared as more paternal genetic materials is introduced, and by the time we hit F5, the phenotypes are indistinguishable from that of the paternal lines, again showing that POE is not due to mitochondrial inheritance. It is known that some wild isolate contain selfish genetic elements active in either sperm or oocyte, causing them to be genetically incompatible. It is then conceivable that genomic incompatibility may be enhanced in the presence of starvations, and it depends on the directions of the crosses, giving rise to POE we observed. However, although we do observe the strains that we use were incompatible to an extent, as indicated by reduced viability in the reciprocal crosses, we did not observe significant difference between the two directions of the cross or and the starvation nutritional status of the parents, and therefore we conclude that genomic incompatibility is not sufficient to explain the strong POE we observed. Finally, we looked into epigenetic inheritance as a potential mechanisms for POE. Epigenetic marks such as histone modifications and DNA methylations undergo extensive erasure in a fertilized embryo. And so for the longest time, it was thought that epigenetic inheritance was not possible. However, it has become clear over the years that small RNA can act as a carrier of epigenetic memory. Lucky for me, we know quite a lot about the mechanisms of small RNA inheritance in C. elegans, thanks to a lot of the work from a lot of people, including Nick, who gave a virtual talk 2 weeks ago. In this model, small RNAs such as piRNA in the germline engage new RNA-dependent RNA polymerase to direct the synthesis of secondary small RNA. And by guiding argonate proteins to the target through the nuclear RNA pathways, the secondary small RNA can then mediate transcriptional changes, in part by causing histone H3K9 methylation. To test if small RNA inheritance can mediate the maintenance of POE, I starved the animals and did the crosses like I did before, and knock down some of this component in the F4 animals that show POE and assay for SKN-1 phenotype in the F5 generation. In the control experiment, POE is robust at F5. That is, the phenotype of the F5 progeny followed that of the maternal founder. Again, the progeny with a high maternal founder has a high phenotype, and progeny with a low maternal founder has a low phenotype. However, disrupting piRNA synthesis erased POE, and the phenotype of the progeny from the two directions of the crosses are no longer different from each other. Likewise, disrupting the nuclear RNA pathways or the H3K9 methyltransferases also erase POE. In conclusion, we found that starvations trigger heritable epigenetic effect regulating gut specification through the piRNA nuclear RNA pathways, which may subsequently lead to repressions of the paternal allele, leading to non-reciprocal POE effect we observed. With that, they proposed that the rapid evolutions of the endoderm GRN we observed among C. elegans wild isolate is driven not only by accumulations of cryptic genetic variants, as I showed you in the first part of my talk, but also selections on phenotypic variation caused by epigenetic inheritance. Lastly, I would like to thank all members from the lab and people who had helped me with this and my other projects, Young La, my mentor, and all my undergraduate research assistants. And I'm happy to take any questions.
Thank you, Ethan. So for asking questions, you can either raise your hand through the participant tab. So you click on participant on the bottom of your screen, and then you can see the raise your hand in the option below the participant name, or you can write the question down in the chat box or into the C. elegans Slack channel. While we're waiting for a question, I have one. So do you have an idea of why you see such variation of like was it SKN-1 expression through the natural populations of C. elegans? Right.
So I think at first we were thinking that it was driven by all the genetic variations that we saw and among the wild isolates. So we did a GWAS, genome-wide association studies, and QTL mapping, and found there indeed genetic variants that may explain it. And then in my second part, I talked about there may be epigenetic components that regulates requirement for SKN-1. Yeah, did that answer your questions? Yeah.
Yeah, it's okay. So we're going to start with the first question from Nick Burton. Great talk, Ethan. Have you tried any other stressor other than starvation, such as a Pseudomonas aeruginosa infection?
No, I haven't. I did. So this all my assay, I use Dower. I starved them as Dower. I did try to starve them as L1s, and that didn't appear. Well, my very super preliminary. I didn't see an effect with L1 starvation. I haven't tried any other stress such as bacterial infections. Yeah, that'll be interesting to do.
And as a second question, if you sequence the small RNA in the progeny of starved animal and might have an idea of what target gene and small RNA might mediate this process.
Yeah, that's definitely on our long list of things to do next. Yeah.
All right. And then we have a question from Yujun Wu. I'm going to unmute you.
Hi, I have a question about the phenotypes of the epigenetic mutants. So in all of your mutants, the POE was lost, but some of the mutants have low percentage of endoderm and others have high percentage of endoderm. Can you explain it?
For this experiment, you said?
Yeah.
Yeah, so you're saying these guys are higher than this ones?
Yeah.
I think it's just natural variations. I haven't, if I've done enough, they probably will bring it all down. Yes, they seem to be a trend, but right now we don't have enough data to show if it's actually true, if it's actually real.
Okay.
Yeah.
Thank you.
Yeah, it's interesting. Yeah.
All right, thank you, Eugene. And a question from Dana Miller. Does it matter how the animals devours? Do you get the same effect if you use a pheromone versus a crowding starvation versus DAF2?
Yeah, we right now I use 2 weeks. 1 week didn't show me an effect, 2 week did. But I certainly haven't tried pheromone and other ways to induce devours. But seems like how long they experience starvations may influence it. Yeah.
All right, thank you very much.